Hello, everybody. Good afternoon. Well, good afternoon here, I guess. It may not be afternoon where you are, but who knows? Anyway, hi, welcome back. Day 31 of our bioinformatics learning for this weird quarantine period that we find ourselves all stuck in. So I hope everybody's well, and we'll get right in. I will just mention briefly that I am aware that the archive of yesterday's video, so the kind of uploaded version of yesterday's video after the live stream, which I tried to edit to get rid of the most of the countdown at the beginning, because having 5 minutes of that music on the video, on the kind of more permanent version of the video, I didn't think was the best thing in the world. But me trimming the first 4 minutes of that countdown off appeared to also trim the last 4 minutes off the video, which I wasn't entirely anticipating. And once you've made that edit on YouTube, then the video that you've trimmed away is gone, apparently, so it seems. So thankfully, I waffled at the end yesterday, as I am prone to do. So there really wasn't any kind of meaningful content in that last 4 minutes. So if you're not following live, you've just kind of tuned back in, having just watched number 30, and now you're watching the beginning of number 31, and you didn't miss anything at the end of number 30. Not anything significant anyway. So what I'm going to do today, rather than trim the video, is I'm going to try and put chapters into the video so that you can skip over the countdown timer to the kind of beginning of the session, and my waffling if you want to. So there you go. Updates, housekeeping updates. The other thing that I just briefly want to mention is that I have now uploaded the beginnings of our script. So the script that we've been writing over the last couple of days, I have now uploaded the beginnings of that script to the Lockdown Learning GitHub. So you can see that the script up until now I have committed there. So that's got reading in the sample table, processing the sample table to get us down to just the 12 rows that we're interested in, and then also making the sample files vector, named vector, and making reading in the gene map as well is there, and then the TX import and the DSeq that we did yesterday. So the DSeq dataset from TX import that we did yesterday is all there. So kind of up to this point in time, all of that code is now committed on GitHub. You may have also noticed I've also uploaded my gene map as well. So my gene map CSV, which is quite a big file, is also up on GitHub, so that if you missed that bit, or if you, for any reason, couldn't get this to work correctly, then you can, again, get my version of that from GitHub so that it's there. Okay, so that is definitely all of our housekeeping. And so what we're going to do today is pick up from where we left off yesterday. So if you recall, yesterday we read our data set into DSeq using the DSeq function, DSeq dataset from TX import, and we also normalized our count data. As we talked about the reasons behind normalization, why we do it, and how it makes our data comparable between samples. And then we ran the normalization using the DSeq method, which was estimate size factors. And I said that what we would do today is talk about PCA. Now, in my prep for today, I have run the PCA for these 12 samples, and I'm going to show you how to do that in a second. It has flagged up one immediate issue, which is a slight spoiler alert here, but it turns out the cell lines, the predominant difference, which is not a surprise in this experiment, is the cell line, the cell type that has been used for what are essentially two experiments that are in this data set. That's a much bigger difference than I thought it was going to be. Much bigger difference than I thought it was going to be. And so what we're going to do, we'll run the PCA, and I'll show you the PCA and show you what I mean by what I'm just saying now. And then we're going to split our data set in two. And we're going to proceed with the analysis on the data split into two separate sub-data sets, because I think that the difference between the cell lines is just too big for us to consider this to be one data set. But I'll show you what I mean by that. Let's do what we need to do to run the PCA. So in our R session, here we are. So we'll just have a quick squiz at the environment and see what we've got so far, because we've not actually got a huge amount, but we've got our count data. That was the results of TX import. We've got our gene map and our sample table as well. And we've got a couple of vectors here we don't need to worry too much about anymore. And then we've got our DSeq dataset, and it's our DSeq dataset that we need to worry about for the next bit. So our DSeq dataset, as we saw yesterday, it now contains our raw count data, and because we've run estimate size factors, it also contains the normalized count data. So the normalized count data in there is so RNA-Seq counts are interesting. They are it's generally agreed they can be modelled according to this statistical distribution called the negative binomial distribution. But what they are not, and again, I think it's a reasonable statement without being a full-on statistician, but what they are not is normally distributed. And principal components analysis, which is what we want to apply at this point, is a parametric technique. It assumes normality. So PCA is a technique that you apply over normally distributed data as much as possible. At least that's one of the assumptions that you make when you run PCA. And our count data, even after normalization, is not normally distributed. But RNA-Seq data is roughly log normally distributed. So if you log transform it, then you get an approximate normal distribution. So we can run PCA over log transformed counts. The problem that we have with that is if you just log transform it, zeros create issues. So you can't apply a logarithmic transformation to a zero. So you have to do a small transformation to the counts to just apply a log transformation. But also just directly applying log to the base 2 across your entire count table is not the best thing in the world to do. It's most of the reason for this is because RNA-Seq data is what's called heteroscedastic, so its variance is not stable across the range of the data. So counts with low counts, so small counts, are highly variable, and high counts have much lower variance, and that heteroscedastic nature of the data means that a normal, just a standard log transformation without taking that into account would have strange effects on the data and would make the variance of the data look a bit weird. So DSeq gives us options for effectively doing a log transformation with our data. There are two options. One is called a regularized log transformation, which takes account of this this changing variance across the scale of the data. The second, but is just a straightforward log transformation. The second transformation is called a variance stabilizing transformation, which is not directly a logarithmic transformation, but results in log scale data, and again deals with this uneven variance across the scale of the data. The primary difference between these two transformations in DSeq, to be honest, is speed. There's not a huge amount of functional difference between the results of the regularized log transform and the variance stabilization transform. So the rlog is slow and the VST is, relatively speaking, fast. So I'm going to apply the variance stabilizing transformation to our data set here. So I'm going to use this to make I'm going to get a new variable at the end of this, which I'm going to call VST, so that I know it's variance stabilized. It's the results of a variance stabilizing transformation, and then the function, which is a DSeq function, you can see it here in my little completion dialog. Variance stabilizing transformation, and that just takes your DSeq dataset as the first argument, and that's all in most cases. That's all you really need to do is pass it a DSeq dataset, and this will apply this transformation to the dataset. So just quickly, what I'm going to show you is, let's have a look at a box plot of the count data, of the normalized count data. So box plot is a base graphics function which will take a matrix and will plot the columns of the matrix as boxes. So that should, and it will take a little while because this is a big data set, but that should show you the kind of distribution of the normalized count data in box plot form, except that, here it goes, it's going to take a while to draw because it's got a large amount of counts, so a large amount of outliers in the boxes, so it's just taking a little while to draw on my machine. But we'll do the same once we've so we're going to run the variance stabilizing transformation, and I'll show you the difference that it makes. So that's our just straight count data, and you can see first of all that we're crossing many orders of magnitude. So we're going from counts of 0 to counts of hundreds of thousands, depending on the gene. And also that, I mean, the bulk of it is squished into this bottom range, sort of between probably 0 and about 1,000. But because the range of the data is so big, you end up with your boxes that look decidedly weird. So we're going to apply the variance stabilizing transformation, and then we'll take a look at the same data once it's in, this data is logarithmically scaled, even if we haven't applied a log transformation. So to get at the data from your VST, you have to use this accessor method, which is called assay. That gives you back a matrix of the data. Actually, let's just put these. I'm typing them in the terminal, but really I should put them in my script. So there you go. We'll make a box plot of the VST, now the VST transformed data, variance stabilization transformation. So we'll have a look at the box plot of that data. And you can see, okay, so there's still quite a large range, although it's from 0 to 20 now, rather than from 0 to 600,000. and that's because it's log scale. So now a change in magnitude of 1 on this scale is a doubling of the data. So despite the fact we range over 20 units, it's still a range of hundreds of thousands in the linear scale. And actually, one thing you can sort of see, you can see it more on the box plot of the counts, is the difference between the first 6 samples and the second 6 samples. There is actually a difference in the overall magnitude of the counts. So the first 6 samples have these very high counts, whereas the second 6 samples aren't exhibiting those very high counts. So you can see that there is a systematic difference, even in just these box plots, between the first set of 6 samples and the second set of 6 samples. That's highlighted all the more in the PCA. So now that we've got this VST dataset, so this transformed dataset, we can now apply principal components analysis to that dataset. And again, DSeq gives us a way of doing this. So DSeq gives us a function which is plot PCA. So plot PCA allows you to create a principal components analysis plot from this VST transform, so this variance stabilizing transformed data. So we need to give this 2 arguments: the data itself, so that's our VST variable that we just created, but also you need to say which column of the col data you want to color your points in by. So which of our columns in our sample table do we want to see represented as the colors on our PCA plot? And you do that by telling it the int group, so the group of interest. And the group of interest here is the conditions. So that's the column that we're in our col data that we're interested in. It's the column that's in our design that we refer to in our design matrix here. Okay, so that's saying, okay, generate me a PCA, a principal components analysis, from this data, from the data in the columns. So I'm interested in the PCA of the samples, and color it in by the variables, by the levels of the factor in conditions. So that's what we're going to do, and this will produce a principal components analysis, which now you can see in my plots window. So I'm just going to make this, I'm just going to zoom to the plots window just so we take up the whole screen with it. Zoom plots, there we go. And this is I showed this to Peter this afternoon because I generated it and it was like, oh, that's interesting and not great. So this is PCA, and I'm not going to go into the maths of PCA because to be honest, like I sort of understand it, but I struggle to explain it. But the main thing for PCA, kind of as a technique to use for a bioinformatician, what we need to understand about PCA is that the closer 2 points are on this plot, the more similar they are to one another. So if 2 points are close together, then those 2 samples, because this is representing the samples in our RNA-Seq experiment, those 2 samples are similar to one another. So these 2 green ones here, for example, are very similar to one another. And then the other thing that we need to understand is that the principal components, which is what this analysis has generated, these principal components represent an amount of variability, an amount of variance in our data. So each principal component explains a proportion of the variance in the data. The thing to note about this is, in this extremely unusual PCA plot, is the first principal component in this PCA explains 98% of the variance in the whole dataset. Although actually, a thing to note about the DSeq PCA plot is it only uses the 500 most variable genes in your dataset. So this is actually explaining, this first principal component is explaining 98% of the variance in those top 500 most differentially most differentially expressed genes. So this is very unusual. You would normally expect the first principal component to explain a lot of the variance, maybe as much as 50 or 60%, sometimes even more than that. But 98% is extremely unusual. And that axis of variation is entirely occupied by the cell line. So on the right, the blue and the red here are both A549 cells, and on the left, the green and the purple are NHBE cells. So that variance, that amount of change is entirely explained by the different cell lines that we've got in the data. So those cell lines are exhibiting, without even considering whether they've been infected with SARS-CoV-2 or not, those cell lines are very, very different to one another, even just in their baseline expression. For this reason, and so then the second principal component, it's saying 0% of the variance, but this is rounded, so it will be less than 0.5% of the variance. This says to me, well, these cell lines are so different from one another. I mean, they are essentially completely different from one another in their expression, that we just can't consider this to be one experiment. That normalizing all of this together makes no sense to me, because the cell lines are completely different. If we try and take account of the effect of the cell line on gene expression in this experiment, we will remove essentially any differential expression that we might observe. So what we're going to do from this point on the basis of this PCA, is we're going to separate this into two separate experiments. So we're going to go back to our DSeq dataset. We're going to chop it in half and have two DSeq datasets, each of 6 samples, each representing one of these cell lines, because it's not a great idea to analyze this all together. So I think that's what we're going to do next, basically. Yeah, this was interesting, and it kind of shows, to a certain extent, it shows the value of PCA, just in terms of very, very rapidly, and it's a rapid technique. It's quick to use in the context of DSeq. It's easy to use because they provide you a method for doing it. We can look later at deriving the principal components ourselves to give us a little bit more flexibility in the plotting that we're doing here. And we can talk about that a little bit later on when we've got back down to one experiment that we want to analyse. But it's a really straightforward technique that we can apply over the top of this data and very quickly get an idea of any really big sort of gross problems that we might find in these data sets. The first thing that this is saying to me is this is 2 experiments, it's not one, so we need to split it out. It's not telling me any more than that, really, because any real variation between the samples is being completely elided by this variation in the cell lines. So I'm not going to talk any more about PCA at this point, other than to kind of use this as the basis to move on to the next step. So I'm just going to unzoom. The only other thing I want to note about this plot PCA function from DSeq is that the plot that it produces is actually produced by the underlying plotting library is ggplot2. So we can treat this plot as a ggplot2 plot, which means we can alter it in the way that we could alter a ggplot2 plot. So what I mean by that is we can change its we can change the way that it looks in the same way as we would a ggplot2 plot. So by adding things to it, so we can add a theme. So we can add theme BW to this plot. Why did that not work? That must have not loaded ggplot2. That's interesting. Oh, because we haven't used it in this script yet. That's why. So I need to load ggplot2. So you'll just note I always put my library commands at the top of my plot. I never ever hide them away in the middle of a script, because I want to, if I come back to this script in 6 months' time, I want to I would want to know, like, straight away, which packages does this script require, what things do I need to make sure that I have in my R session, so that this script will work. And so I always have my package loads at the top of a script. So to go back to this, sorry, so we can apply a ggplot theme to the results of our plot PCA in exactly the same way as we would do if we generated it ourselves using ggplot. So that's quite a useful thing to be able to do. It lets us do things like add labels to the plot and things like that when we want to later on. Right, okay, so I'm just gonna make a comment in here. Observe the cell line effect, and then we're going to split the experiment in two. So to do this, so we've got, at the moment we have our DE-Seq dataset, which is called DE-Seq dataset. I'm going to split it in two. So I want DDS1, so DE-Seq dataset 1, which is going to be, we can treat, for the purposes of splitting it up like this, we can treat it as though it's a data frame, and we want to subset the columns because we're interested in subsetting on the samples. So we want, in our first data set, we want the first 6 columns. So this will give us, if we just, at the console, if we just print our DE-Seq dataset, we get a little summary of the dataset. So it tells us the dimensions here. So 60,000 rows and 12 columns. After we've subsetted like this, we should see that our dataset DDS1 has 60,000, still has 60,000 rows, but will only have 6 columns. And it will have GSM blah 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 378, 379, 80, 81, 82, 83, and 84. Okay, so let's do that. That's the first data set. Let's have a look at DDS1. So there you go, 60,000 rows and 6 columns, not 6 columns, not 12, and 6. There are your column names up to 8.3, not 8.4. Can't count. And then DDS2, the other half of the data, we want to be, we're going to split in exactly the same way. So we're going to split our DE-Seq dataset on the columns, so after the comma in the square brackets, because the rows are always before the comma, and the samples are always, the columns are always after the comma. And we want columns 7 through 12 in our second data set. Okay, so that's splitting the data set up is as simple as that. But when you subset the data like this, we have to redo the normalization, because the normalization that you get for your whole data set is a cross sample. So the normalization of one sample depends on the presence of all of the other samples. So if you change the set of samples that you have in your data set, then the normalization also has to change. So when we split this data set out, we've passed the normalization factors across from when we normalized with all 12 samples all in the data set. But we don't have all 12 samples all in the data set anymore. So the normalization is going to change. So we have to recalculate those normalization factors because we have now resubsetted the data. So we have to go back and recalculate that normalization. So we have to do it separately for each of our data sets. So we now have two data sets, one called DDS1, which we want to normalize using estimate size factors, DDS1, and the same for DDS2. Okay, so again, recalculate those size factors for DDS2, those normalization factors for DDS2. So if we just quickly look at the normalization factors for the first 6 genes and the first 3 sorry, that's not what I wanted. Sorry, the normalization factors in the original data set for the first 6 genes and the first 3 samples. So those are those normalization factors in that original data set. If we look in DDS1, which does contain these same 3 samples, we will see that those normalization factors are different. So it is important. They're not massively different, but they are different. So it is important to go back in and recalculate those normalization factors after you've split your data up, because the normalization factors have to be based on the data that you have. And so I should say the raw counts are still the same. So the thing about this DSeq data set that we're working with here is when you calculate your normalization, what you're doing is the DSeq data set is this complicated object. It's essentially a big list, really, but it's a complicated data structure. And we're not overwriting the raw counts. So the raw counts for so if we do counts DESeq dataset, again we're going to do 1 to 6 and 1 to 3. So the top left hand corner of the dataset. And if we do normalized equals false, so that's the raw counts. Those raw counts will be the same in DDS2, in DDS1. So if we look at the same function call, the same rows and columns in DDS1, you'll see that the raw counts are exactly the same. And the normalized counts are calculated by applying the normalization factor to the raw counts. So we're not, as somebody in the comments suggested, we're not normalizing already normalized data. We're just calculating new normalization factors and applying them to the same raw data. So the normalized data, we're not renormalizing already normalized data. We're just recalculating different normalization factors and applying that new normalization factor to the same raw data. So the normalized counts are different because the normalization factors are different, not because we've renormalized already normalized data. And if you want to do the calculation, you can. So you can do it's hold on, let me get it right. DDS1 slightly larger than 1, so it must be yeah, okay, so let's do this. It's the no, it's normalization factors. Normalization factors of DDS1. Okay, and again, do 1 to 6, 1 to 3. So, damn it. Hold on, I'm going to type that again now. Right, so counts. So we can derive the normalized counts, is my point. So if we take the raw counts and we divide them by the normalization factors from the same data set, and then we take the same slice of data that we've been taking all the way along. Why does that not work? Should work. Anyway, if we go up to the top corner, they should be the same. They are the same, it's just really hard to show, sorry. Let's do as.matrix. Check that this works. Sorry, I just want to check that this works before we move on. I'm just distracting myself. There you go. So, and again, if we get counts DDS1 normalized equals true, 1 to 6, 1 to 3, you should see that the numbers are the same, right? So what DSeq is actually doing when you put normalized is true in here is it's doing this division that I've done here, so dividing the raw counts by the normalization factors to give you back the normalized counts. So the DSeq dataset is a really clever thing, and this is, I haven't talked about the way that R encapsulates this more complicated data yet, but the DSeq dataset is a really clever way of encapsulating this data so that we can keep updating it and we don't lose what we had there before. We've not overwritten the raw counts. We've just added information into our data set. So I'm going to redo the PCA, and I'm just going to do it with DDS1 now. So to redo the PCA, we need to redo the variance stabilizing transformation. So we're going to do VST1 equals variance stabilizing transformation DDS1, like that. Shouldn't take very long. And then we want to plot PCA VST1, and again the int group is conditions, which is the column in our design matrix, which is referring to these conditions. Okay, so I didn't get as far this afternoon as doing this bit, so I don't know what this is going to look like. So I've got no spoilers to give you at this point. Okay, this looks more like a normal principal components analysis plot. So somebody asked before, what would it look like if there wasn't such a big cell line effect? It would look something like this. Okay, so we've got, we're seeing here two principal components. One which represents just over 40% of the variance, and one about 20% of the variance. And on that first axis, we're really seeing the variance that is due to the SARS-CoV-2 infection. So the infection is affecting gene expression. We know that. We can see that in this plot. And the other thing that we can see is that the mock-infected samples are a bit more variable than the infected samples. So the mock-infected samples have a wider spread on our PCA plot. So they're a bit more variable than the infected cells, which is fine. It shouldn't make much difference. The mock, you can still draw a circle around the mock samples on this plot without encapsulating any of the infected samples. So I would be pretty confident that we can analyze this data and get differentially expressed genes out of it. And hopefully that's what we're going to do. I think we're going to do that tomorrow. I'm not going to launch into that now. We will look at deriving differentially expressed genes from this data tomorrow. So that's good to see. Let's have a look at the others as well. Let's see if the other cell line behaves as nicely as this one. We need to do another variant stabilizing transformation, and then do another PCA plot. And I'm just going to copy and paste my code and then change the 1s for 2s, because that's the fastest way to do it. Okay, here we go. So that's running the VST and then the PCA, and there we go. Very kind of pretty similar story, really, in the A549 cells as well. So again, the primary axis of variation is mostly the infection, it's mostly the presence of the virus, and then the second is again the variation in the mock infected cells, with a little bit more variation in the actually infected cells this time. So that's quite a nice observation. So both of these experiments behave quite nicely, and we should, from both of these experiments, be able to hopefully derive some differentially expressed genes. I just want to think a little bit more about sample distance while we're here. So PCA is one mechanism for showing you, in a pretty non-quantitative way, the difference between samples. So there's no, although we can see how much variance each of these principal components accounts for, and so you do get some sort of an idea of the difference between samples here, and if they're more separated on the x-axis, that means more than if they're more separated on the y-axis, because that's a larger amount of variation. But it's only one way of looking at how related samples are to one another. There's lots of other ways as well. Something else that people often do is some sort of clustering to see if you can find groups in your data. So there are ways of doing supervised clustering and unsupervised clustering, and we can look at those as well. So we can do, R has very efficient and very robust methods for doing these kind of clustering operations, because they're statistical operations and R is built for statistics, and they're functions of the base libraries, because again, because this is R is a statistical package. So I'm just going to show you a couple of examples of other types of clustering. So just to show that it's possible. So I'll do it with VST1. So Again, we're going to use our VST data because it's more normally distributed, so it's more likely to be amenable to these kind of techniques. And we're going to do, to begin with, we're going to do hierarchical clustering. So there is a method for hierarchical clustering in base R, which is hclust. So hclust runs hierarchical clustering over an input matrix, but the input matrix that it needs is a distance matrix, not a so it needs a distance matrix rather than a matrix of observations. So our matrix of observations is in this VST1 object, and we have to get that matrix back out using the assay function that I talked about before. So assay here for VST data is achieving the same thing as counts is for our DSeq dataset. So if I do DDS1 just on its own, if I just print DDS1 in my console, you will see that it just prints out this summary of the data. In order to actually get at the count matrix, I have to use the counts method to do that. And this is because of the nature of the dataset. So that gets me the counts matrix. And the VST, this VST object that we've created here behaves in the same way. It's what's called a summarized experiment. And again, if I just print out the dataset itself, it's more than just the matrix. It's got more things in it than just the matrix. It's got column names and row names, and it's got the col data in there as well. It's inherited the col data from our DSeq dataset. To actually get at the data that's in there, the VST transformed data, we have to use, like we have to use the count successor method for our DSeq dataset, for our VST dataset, we need to use this assay accessor method, and that gives us back those variance stabilizing transformed counts. So this is the counts data after that transformation. So in order to get at that matrix, we need to use that method. But this is not the input for hclust. The input for hclust is a distance matrix, and we can calculate a distance matrix from this expression matrix using again a base method from R, which is called dist. So dist will calculate the distance between the rows of your dataset. So it works across the rows, and our samples are in the columns. So we want to know the distance between the samples in our dataset. And to know that, we need to, in order to know that, we need to transpose the matrix. So we've looked at this T function before for transposing a matrix. So I've nested these functions here. I'm going to pull these out of this nest because I think that's particularly a particularly confusing thing for beginners. So let's pull this out of the nesting. So let's say, okay, we want a distance matrix. So we want a thing called D, which is a distance matrix. So to begin with, we're going to get the expression data, VST1. So the assay data from VST1. Then we're going to transform that. So I'm just going to recycle my variable name. So we're going to transform D, transpose D. And then from that transposed expression data, we want to calculate a distance matrix. So we want to calculate the distance matrix of D using this function dist. Dist, by the way, and it's kind of beyond the scope of what I want to talk about today, but dist lets you calculate a distance matrix using lots of different methods. So lots of ways of calculating distance: Euclidean, maximum, Manhattan, Canberra, binary, Minkowski. My colleague John does a really good, he's got a really good talk about the differences between these different distance metrics, which I'm not going to attempt to recapitulate. John's a mathematician and I'm not, because maybe one day if my YouTube ambitions get any greater, I will invite John on as a guest to give his talk about distance measures and about clustering. But for today, what we need to know is we've done a certain amount of evaluation of these distance methods, and Euclidean for expression data. Euclidean distance is almost always the best distance metric, and Euclidean distance is the default, so we don't need to change it. So this will calculate the Euclidean distance between our samples. Let's get rid of that. And then that gives us our distance matrix, which is our input for our hierarchical clustering. So we're going to stick our distance matrix in there. And again, hclust has different ways of doing the clustering, different methods for doing the clustering. So again, like dist, hclust has a method argument, which by default is complete, but could be any of these other versions as well. We're just going to use complete clustering because it's fine. It will produce us what we want. So again, we're going to use the default, so we don't have to do anything here. So I'm just going to save this just as a variable called h. Okay, so we're going to calculate our distance matrix using dist, and then we're going to pass that distance matrix into the hierarchical clustering function to generate h, and then just using base plot, we're going to plot h. So we're going to just run plot over the results of our hierarchical clustering, and that will produce for us a cluster dendrogram. There are fancier ways of producing a dendrogram that will produce you much nicer looking dendrograms than this, but as a first pass at this data, this is quite a nice. This is perfectly fine, right? There's nothing wrong with this, so this dendrogram is generated from the results of the hierarchical clustering. So hierarchical clustering is an unsupervised method of clustering. So the clusters in the tree, they could be any size. They could have any number of members. You can cut this tree in multiple places and generate lots of different numbers of clusters. So you could cut this up at the top here and generate 2 clusters, which is probably the most natural way of doing it. But you could cut it down here and chop it up into 4 clusters, 2 of 2 and 2 of 1, for instance. Because it's unsupervised, that you don't predetermine that up front. There is a way of doing supervised clustering with this data, of saying, okay, here are the number of clusters I expect, and please can you separate my data into that number of clusters. The way that you would usually do that is by k-means clustering. And again, there's a base function for doing k-means clustering in R. I'm just trying to figure out if it takes the distance matrix or if it takes the actual data. I think it might take the actual data. We can only really know by trying. Okay, so k-means is a method called k-means, which takes a numeric matrix of data and the number of centers, that's the number of clusters that you want to generate. So this k-means is supervised clustering. So you tell it up front how many clusters you want. I think, again, this will cluster on the rows, not on the columns. So we're going to use our transposed matrix of data as input, like that, and we know that we've got 2 clusters. Okay, so we want 2 centers. We're gonna save this as a variable k. hope that works. I've not done this for a long time, which is why I'm unsure. No, right, okay. That has clustered our genes into 2 clusters by the looks of it. So we need to not transpose that matrix. I'm amazed that I got away with that. Okay, so let's not transpose the matrix then. Like that. Hopefully this is a bit more understandable this time. Oh no, that's... what's going on here? Okay, now this really has clustered. Okay. Sorry, I've not done k-means clustering for a while, can you tell? Okay, we do need to transpose it, I think. So what's in K? Let's have a look at K. It's a large k-means object with 5 elements. So cluster is the thing that we're interested in. And cluster, so K, it's a list. The output of this is a list. So you can use the string operator to pull out just the bit of it that you're interested in. So you don't get a visual result from k-means like you do with hierarchical clustering. You just get each of your samples assigned into one of your k clusters, where k is the number of centers that you give the data. So here you can see these are our clusters as determined by k-means clustering. So the first 3 samples are in cluster 1, and the second 3 samples are in cluster 2. So that has partitioned the data exactly as we would have expected it to. Yes, our 3 mock infected samples fall into one cluster, and our 3 properly infected samples fall into the other. So that's just a couple. I'm sorry, it got a little bit improvisy towards the end there, but there's a couple of ideas there of how we can start to think about the relationships between the samples in this data, and how to observe the relationships between the samples in this data. We can go into more detail about how to do these things and how to prettify the output and things like that. I think it's nice just to see a dendrogram. Actually, I think that's quite a nice output, and it's quite nice to see a PCA plot as well. What I haven't talked about today, because it doesn't make a lot of sense when you've only got 6 samples, is the kind of the really high dimension methods which are used for things like single cell data and flow cytometry data. So non-parametric methods like t-SNE or UMAP that are increasingly used for this kind of dimensionality reduction in experiments with a much larger number of dimensions. So as I said, particularly applies to, in RNA-Seq, particularly applies to things like single cell data sets. It is possible to apply those to bulk data sets like this with a small number of samples, but actually PCA it does the job just fine and makes a lot of sense. Okay, I'm gonna leave it there for today. So join me again next time for number 32. That will be tomorrow, so it'll be Thursday. To remind you, I'm not going to be online this Friday. It's a public holiday in the UK, so there will be no lockdown loading on Friday this week. But I will see you tomorrow, which is Thursday, so I will be online tomorrow. Okay, so I'm going to sign off. I will see you all next time. Stay safe, keep well. See you later. Bye.